So it tells us the gene represented in this diagram below that codes for multiple proteins due to alternative RNA splicing. You've got your coding regions shown in black here, that the numbers are above. So they're labelled from 1 to 10, and the white bits in between, those would be the non-coding regions. So it tells us below that alternative RNA splicing results in a mRNA that contains exon 2 or 3. It also contains exon 4, 5 or 6, and it also contains exon 7 or exon 8. And what you need to do is you need to predict the total number of different proteins that can be produced from this gene above, using this information here about the different exons that are going to be contained within each of the proteins that can be produced. So this is the key bit of information here. It tells us that alternative RNA splicing results in mRNA that's going to have to contain either exon 2 or exon 3. So we'll cover that one red. So it's every protein that's produced is going to need to have either one of these, 2 or 3. The next bit here tells us that it's going to need to contain exon 4, 5 or 6. It needs to contain one of these exons. So we'll colour that one blue. And the very last bit tells us that it's also going to need to contain exon 7 or exon 8. And we'll colour that in green. So every possible combination is going to need to contain either exon 2 or exon 3. It's also going to need to contain either exon 4, exon 5 or exon 6. And it's going to need to contain exon 7 or exon 8. So from each of these colours, there would be one exon possible in each combination. You can only take one from each of the colours, but they have to contain one from each. So if we use the information just now and just look at the possibilities that can be produced, the first one we'll look at, we could have one that contains 2, one that contains 4, and one that contains 7. So 2, 4 and 7, that's one possible combination. You could also get a combination that's 2, 5 and 7. And you could also get a combination that's 2, 6 and 7. So you could, as well as getting ones that contain those, you could also get one that contains 2, 4 and 8. So you get 2, 4 and 8. You could get 2, 5 and 8. And you could get 2, 6 and 8. So that's all the possible combinations that contain 2. But you could have exon 2 or you could have exon 3 in your combinations. So here you could look at, instead of being 2, 4 and 7, you could also have 3, 4 and 7. You could also get 3, 5 and 7, 3, 6 and 7, and you could also get 3, 4 and 8, 3, 5 and 8, 3, 6 and 8. Okay, so this here, these are all the possible combinations that you can get for 2, these are all the possible combinations that you can get for 3, and together it's all the possible combinations that are available. So you'll see that for each one, there's 1, 2, 3, 4, 5, 6, so there's 6 for 2, and there's also 6 for 3 available. So there's 6 different combinations for 2, 6 different combinations for 3. If you add 6 and 6 together, you're going to get 12. The original question, predict the total number of different proteins that can be produced from this original gene. As we just worked out, the answer would be C, 12.